Today we are giving an introduction to the book Vertebrates, Comparative Anatomy, Function, Evolution by Kenneth B. Kardong, 8th edition, the International Student Edition for use outside of the United States, published in 2020. We have compared different historical predecessors, comparative vertebrate morphology, talking about the process behind the change, Carl Linnaeus, naturalists like J.B. Lamarck, natural selection, and A.R. Wallace, Charles Darwin, and the historical predecessors of morphology like Georges Cuvier and Richard Owen. Now moving on to morphological concepts, similarities, symmetry, and segmentation, before moving on to evolutionary morphology, phylogeny, paleontology, and then we will finally conclude by a reading of chapter 16, which covers the nervous system. Morphological concepts. To analyze design, concepts of form, function, and evolution have developed. Some of the most useful of these address similarity, symmetry, and segmentation. In different organisms, corresponding parts may be considered similar to each other by three criteria: ancestry, function, and appearance. The term homology applies to two or more features that share a common ancestry, the term analogy to features with a similar function, and the term homoplasy to features that simply look alike. These terms date back to the 19th century, but gained their current meanings after Darwin established the theory of common descent. More formally, features in two or more species are homologous, when they can be traced back in time to the same feature in a common ancestor. The bird's wing and the mole's arm, for example, are homologous forelimbs tracing their common ancestry to reptiles. Homology recognizes similarity based upon common origin. A special case of homology is serial homology, which means similarity between successively repeated parts in the same organism. The chain of vertebrae in the backbone, the several gill arches, or the successive muscle segments along the body are examples of this. Analogous structures perform similar functions, but they may or may not have similar ancestry. Wings of bats and bees function in flight, but neither structure can be traced to a similar part in a common ancestor. On the other hand, turtle and dolphin forelimbs function as paddles, analogy, and can be traced historically back to a common source, homology. Analogy recognizes similarity based upon similar function. Homoplastic structures look alike and may or may not be homologous or analogous. In addition to sharing a common origin, homology, and function, analogy, turtle and dolphin flippers also look superficially similar. They are homoplastic. The most obvious examples of homoplasy come from mimicry or camouflage, where an organism is in part designed to conceal its presence by resembling something unattractive. Some insects have wings shaped and sculptured like leaves. Such wings function in flight, not in photosynthesis. They do not or are not analogous to leaves, and certainly such parts share no common ancestor. They are not homologous to leaves, but outwardly they have a similar appearance to leaves. They are homoplastic. Keep in mind that the similarities are not exact, point by point, but represent overall similarities in appearance. Such simple definitions of similarities have not been won easily. Historically speaking, morphology has struggled to clarify the basis of structural similarities. Before Charles Darwin, biology was under the influence of idealistic morphology, the view that each organism and each part of an organism outwardly expressed an underlying plan. Morphologists, however, looked for the essence or ideal type behind the structure. The explanation offered for this ideal was the unity of plan. Owen proposed that archetypes were the underlying source for an animal's features. Homology for Owen meant comparison to the archetype, not to other adjacent body parts and not to common ancestors. Serial homology meant something different as well, based again on this invisible archetype. But Darwinian evolution changed this by bringing an explanation for similarities, namely common descent. Analogy, homology, and homoplasy are each separate contributors to biological design. Dolphins and bats live quite different lives, yet within their designs we can find fundamental likenesses. Hair, at least some, mammary glands, similarities of teeth and skeleton. These features are shared by both because both are mammals with a distinct but common ancestry. Dolphins and ichthyosaurs belong to quite different vertebrate ancestries, yet they share certain likenesses, flippers in place of arms and legs and streamlined bodies. These features appear in both because both are designed to meet the common hydrodynamic demands of life in open marine waters. In this example, convergence of design to meet common environmental demands helps account for likenesses of some locomotor features. On the other hand, the webbed hind feet of gliding frogs and penguins have little to do with common ancestry. They are not closely related, or with the common environment demands. The frogs glide in the air, the penguins swim in the water. Thus, structural similarity can arise in several ways. Similar function and similar habitats can produce convergence of form, analogy. Common historical ancestry can carry forward shared and similar structure to descendants, homology. Occasionally, accidents or incidental events can lead to parts that simply look alike, homoplasy. In explaining design, we can invoke one, two, or all three factors in combination, analogy, homology, and homoplasy. To understand design, we need to recognize the possible contribution of each factor separately. Symmetry. Symmetry describes the way in which an animal's body meets the surrounding environment. Radial symmetry refers to a body that is laid out equally from a central axis, so that any of several planes passing through the center divides the animal into equal or mirrored halves. Invertebrates, such as jellyfishes, sea urchins, and sea anemones, provide examples. With bilateral symmetry, only the mid-sagittal plane divides the body into two mirrored images, left and right, like the brain of the human and other mammals. Body regions are described by several terms. Anterior refers to the head end, cranial, posterior to the tail, caudal, and dorsal to the back, and ventral to the belly or front. The midline of the body is medial, the sides are lateral. An attached appendage has a region distal or farthest and proximal closest to the body. The pectoral region or chest supports the forelimbs. The pelvic region refers to hips supporting the hind limbs. A frontal plane or conodal plane divides a bilateral body into dorsal and ventral sections. A sagittal plane splits it into left and right portions, and a transverse plane separates it into anterior and posterior portions. Because humans carry the body upright and walk with the belly forward, the terms superior and inferior generally replace the terms anterior and posterior, respectively, in medical anatomy. Like many terms used only in the descriptive anatomy of humans, superior and inferior are poor ones to employ in general comparative research because few animals other than humans walk upright or are bipedal. If you venture into the study of human anatomy, you can expect to meet such specialized terms. Segmentation. A body or structure built of repeating or duplicated sections is segmented. Each repeated section is referred to as a segment or metamer, and the process that divides a body into duplicated sections is called segmentation or metamerism. The backbone, composed of repeating vertebrae, is a segmental structure, so it is the lateral body musculature of fish that is built from repeating sections of muscle. Not all body segmentation is the same. To understand design based upon segmentation, we need to turn our attention to invertebrates, even though this book is about vertebrates. Among some invertebrates, segmentation is the basis for amplifying reproductive output. In tapeworms, for example, the body begins with the head, the scolex, followed by duplicated sections called proglottids. Each section is a self-contained reproductive factory housing complete male and female reproductive organs. The more sections, the more factories, and the more eggs and sperm produced. Some overall body unity is established by simple but continuous nerve cords and excretory canals that run from segment to segment. Other than this, each segment is semi-autonomous, a way to replicate sex organs and boost overall reproductive output, which is quite unlike segmentation found in other animals. Annelids, such as earthworms and leeches, have segmented bodies that provide support and locomotion rather than reproduction. This segmentation differs from that of tapeworms because the annelid body is fluid-filled and forms a hydrostatic skeleton. The hydrostatic skeleton is one of two basic types of supportive systems found in animals. The other supportive system we see in animals is a rigid skeleton. We are familiar with a rigid skeleton because our bones and cartilage constitute such a system. Another example is the chitinous outer skeletons of arthropods such as crabs, lobsters, and insects. Rigid skeletons are efficient systems of levers that allow selective muscle use to produce movement. And although hydrostatic skeletons are perhaps less familiar to you, they are common among animals. And so, as the term hydro suggests, this supportive system includes a fluid-filled cavity enclosed with a membrane. A hydrostatic skeleton usually is further encased within a muscular coat. And at its simplest, the muscular coat is composed of circular and longitudinal bands of muscle fibers. Movement is accomplished by controlled muscle deformation of the hydrostatic skeleton. In burrowing or crawling animals, movement is usually based on peristaltic waves produced in the body wall. Swimming motions are based on sinusoidal waves of the body. The advantage of a hydrostatic skeleton is the relatively simple coordination. Only two sets of muscles, circular and longitudinal, are required. Consequently, the nervous system of animals with hydrostatic systems is usually simple as well. The disadvantage is that any local movement necessarily involves the entire body. Because the fluid-filled cavity extends through the entire body, muscle forces developed in one region are transmitted through the fluid to the entire animal. Thus, even when movement is localized, muscles throughout the body must be deployed in order to control the hydrostatic skeleton. In truly segmented animals, septa sequentially subdivide the hydrostatic skeleton into a series of internal compartments. As a consequence of compartmentalization, the body musculature is also segmented, and in turn the nerve and blood supplied to the musculature are segmentally arranged as well. The locomotor advantage is that such segmentation allows for more localized muscle control and localized changes in shape. For instance, the segmented body of an earthworm is capable of localized movement. Segmentation among vertebrates is less extensive than that of segmentation among invertebrates. Lateral body musculature is laid out in segmental blocks, and nerves and blood vessels supplying it follow this segmental pattern. But segmentation goes no deeper. The viscera are not repeated units, and the body cavity is not serial compartmentalized. Locomotion is provided by a rigid skeleton, and the vertebral column, or the notochord, is served by segmental body musculature. However, segmentation of the outer body musculature does not extend inward to the column and the viscera. Although the vertebrate body is not composed of a hydrostatic skeleton, selected organs are based on the principle of hydrostatic support. The notochord, for instance, as we have said, the vertebral column, contains a core of the fluid engorged cells tightly wrapped in a sheath of fibrous connective tissue. This incomprehensible but flexible rod is a hydrostatic organ that functions to keep the body at a constant length. The penis, for example, is another example of a hydrostatic organ. When properly stimulated, cavities within the penis fill firmly with fluid, in this case with blood, in order to give the penis an erect rigidity of some functional significance. Next, we will discuss evolutionary morphology before moving on to phylogeny.